Okay, my friends, prepare yourself, because I'm going back about 10 years ago to when I wrote this paper called Academia's Dirty Little Illegal Secret. And now, is it illegal or not illegal, what academia is doing to their students? And what I'm going to point to is fiduciary obligations to work in the best interest of the ones that trust you, which are the students. Do the teachers have a legal responsibility to work in the students' best interest? I say they do. There is some debate about this, but let me see what I wrote back in August 2015. This is after about 5 years of extreme frustration with academia. Now, this is getting into a legal situation, and it should be legal, because I'll show you why it should be legal. Let's go with that. Okay, my friends, this goes back to the days of yesterday here, about 10 years ago or so. I just became so frustrated with the academic system that I started to sort of attack. And this was, I put on LinkedIn. I don't use that anymore, so don't bother going up there. Academia's dirty little illegal secret. Now, I wanted to put this into a legal realm. Now, I know everybody makes mistakes. There is no question whatsoever. Everybody makes mistakes. It just happens. So you can't hold somebody legal for making a mistake. But you can hold them legal for knowing that they're wrong and still making that mistake, working in the disinterest of the student, not working in the interest of the student. There's something called fiduciary responsibility. Now let's just dig into this, because let me read to you what I wrote about it, because it was giants and it related to myths and religions and aliens and giants, and it just blew them right out of the water. And it still does. They've kept me completely off the page for 15 years. Okay, I guess the best way to do this is just to read to you what I wrote back in August of 2015. And I said, alternative fossil research collaboration is not happening. I understand in academia, if you're unique and thoughtful of alternative theories like aliens, giants, religions, or myth, you'll be thrown from the scientific nest. You're out. The people that throw you are the dismissive, arrogant, trusted educators. They don't look at the evidence. We trust them, but they don't look. All right? And do they have an obligation to look at evidence? And I'm not talking about just looking at theories. I'm talking about evidence. Now, those that think of other nests, which is anybody that's in as an educator, they think about things outside of what they've been told to say, they're going to be set adrift. They will be shunned. And there's no wings that's strong enough to fly back to that nest. You're done. You're out. You're never coming back. The result is that the research bullies, and these are bullies, control the careers of real scientists, the ones that really want to look. They control their careers. They say, if you look there, you're done. Go ahead and look, and you're out. The result is that the research bullies control the career of real scientists. Students and society loses out. We lose out, and I'm going to show you we lost everything forever. The real open-minded scientists are understandably restricted from understanding the past. You can't open your mind and look back at what is physical evidence. They won't allow it. They are not allowed to do alternative research. All right? No amount of physical or analytical evidence will convince these bullies. And that's a fact. They can't convince the bullies to so much as look at the evidence disputing their publications and accepted theories. Now, I have DNA, I have CAT scans, I have specimens, I have everything, I have chemistry, I have the process down, and it was all totally rejected, and they wouldn't look at it. So the students lose out. So the students being the fiduciaries, I mean, the fiduciaries are the schools. Their most important thing is to look out for their students, to take care of their students who are paying them tons of money to learn the truth. And these knowing that the truth is not what they're teaching, that's a legal failure. A fiduciary, if they fail to act in the best interest of their trust, people to trust in them, then they're in legal jeopardy. Let's just go with that. Now, let's just continue on. There's no evidence you can present they'll even look at. They're quite comfortable in completely, arrogantly dismissing my voluminous physical testable specimens. I have CAT scans. I have DNA lab reports, and I recreated a process that accounts for the mud fossil chemistry, as well as expert structural anatomists agree with the things that I am presenting. They're biological. And a DNA that shows the matter was alive at one time. All this stuff was done. This was in 2015, done. Even with this supporting evidence and physical specimens, they remain unexamined at this point by academia experts. They still won't look at it. They refuse. These are mud fossils. This is what I discovered 15 years ago. They are not understood or studied yet, except by me. I'm the only one doing it. And here's the CAT scans of a large primate, which was these giants. This is what freaked them out. I presented all my evidence by videos and pictures to Yale, Harvard, and dozens of more. Now, Yale spoke to me and pretty much treated me like I was insane, although they did sign off eventually saying, yes, Roger was right. And here's their paper, Exceptional Preservation of Soft-Bodied Creatures, promoted by Silica-Rich Oceans. This paper basically says exactly what I said. And this is from 2016, a year after. And the only reason I can figure they did this was because they got funded by NASA, who I also contacted about mud fossils. And then apparently somehow they ended up getting to Yale and asked Yale to do this, or well, I don't know how that happened. I can't say one way or the other. But I do know that NASA paid them to do this, funded them. Exceptional preservation of soft-bodied creatures. That's exactly what it is. And it was in the Triassic. My stuff is Triassic too. So this is why I was so upset. All right, no, they would not allow me to bring them in for them to examine them. Even with the DNA test, the CAT scans, all that stuff. So Yale just declared me basically crazy, and they were just simple rocks, not fossils at all. And then they did take credit for the exceptional preservation of these soft tissues. I don't think that's right. Harvard was polite, but they were just dismissive, disinterested, refused to look into any of this and just stay away from us. You know what they did? They said, why don't you go to Yale? You're right down the street from Yale. I said, I did. I showed these things to Derek Briggs. I sent them all the evidence. They refused to look at them. And they said, well, obviously, because you're an idiot. I mean, I don't think they said that in those words, but that was the tone of the discussion. Well, if they won't look at them, why would we? And that's what everybody said. They said, you're right down the street from Yale. If they won't look at them, we're not going to look at them. So once Yale decided, without even a look, they were rocks, it's hard to convince others Yale is wrong. They're the big shots. And I went to the top guy at Yale, Derek Briggs, who is one of the authors on that paper I just showed you. I was persistent, and they have gone dark long ago now. Derek Briggs was talking to me for a while, and then finally he just said, you talk to my assistant, Armand. Armand, you remember these words, Armand? I told you when this is over, you are going to look like fools. And Armand's last words to me were, well, we won't be the only ones. And you are right, Armand. Academia is a disgrace. I was persistent. They went dark. Lack of accountability in academia leads to behaviors not tolerated in business. If I was trying to sell something and I knew it was a fraud, they'd put me in jail. I've been deeply involved in this research for about 3 years and made many scores of communication attempts to everybody, including NASA, including Yale, including Harvard, including everybody. I have not received a single in capitals, response. I'm saying not an email, not a phone call, and have been hung up on at the mention of my research. Just a click. And now my name, as I have been systematically shunned. I'm done. I understand their fear, but they are accountable, although few know it. These are educators. They are educators. That's their duty. Teaching our children what to believe for big money. A fiduciary, this is in quotes, a fiduciary duty is an ethical and moral obligation to act in the best interest of someone that pays you and then puts their complete faith and trust that you will act in their best interest. That's what a fiduciary is. This is a legal duty with financial remedies. Right? I would want my money back. Is there a lawyer in the house? And no lawyers will do it either, because you can't go up against people like Yale, and they'll just destroy you too. Academic research's dirty little illegal secret. I am about to present my opinions and evidence about the state of research in the USA. And now this goes back 10 years ago. A fiduciary duty, ethical and moral obligation to act in the best interest of someone that pays you and then puts their complete faith and trust that you act in their best interest. All right? These are, few realize educators have this legal obligation. Now, I believe it's a legal obligation. They could say, oh, well, we could teach them anything we want. But they're paying them and putting their faith and trust in them. And if they won't look at something that shows that they're not teaching them the correct stuff, they've lost their faith and trust. And they keep their money. And if they don't say what they tell them to say, they tell everybody else that these people are failures. This is the worst possible situation that could possibly exist. These are obligations to the most vulnerable. These kids are the most vulnerable. Students are at the very top of that list. Not only must they trust, but they must learn and agree. You have to agree to be considered educated. And when I say learn, they have to read and keep repeating what they're being told. And then they have to agree with it. They can't say, well, what about this? Believe me, you start making any kind of commotion, you're done. All right? Any opposing views can mean a poor quality of life and a poor grade, yes. The guy wouldn't listen. He didn't say the same things we told him to say. He's got to be stupid. All right? It's a success or failure relationship, and subservience is required. You have to say, yes, massa, I'll do what you tell me to do. And that's exactly what is going on right now. It's a master-slave relationship. Fiduciary obligations from educators to students are implicit, obvious, and never more appropriate. It's just completely obvious. Gross neglect of these legal obligations cries out for refunds. In business, that's exactly the remedy. These are businesses. Make no mistake about it. Every one of these schools is a business, a big business. Hold on. All right, students come to be taught the truth so their lives will be better. This education is very expensive, and debt itself makes their life worse. And now there's all kinds of, this goes back 10 years, but now they're saying they want to relieve the student debt. Yes, they should, but just colleges should pay them back, not us, not the taxpayers. These places that took advantage of the kids didn't do their jobs and keeping their money, and now we have to pay the kids? I don't think so. Go for the schools. The students expect to get what they're paying for. Students naturally and rightfully expect each school and professor to be a good fiduciary and up on the latest information, including CAT scans, DNA specimens of giants and perfectly preserved human fossils, just like this lung that boiled literally out of a human creature, because it's human DNA, and it's flat as a pancake. It was in the flood, and it was a hot water flood, and I have all the information about this, and Velikovsky did too, and he was also destroyed by academia. Same people, same bullies. All right? The better the school is claimed to be implies more research into new discoveries and vigorous scrutiny of these new things naturally is expected. Obviously, you have to scrutinize. You can't just say, get away, go away, go away. And that's what happened. Students expect their trusted professor to have done their homework. This would require thoughtful examination of physical evidence. I'm not asking you to say, oh, I'm saying the earth is flat, and I want you to study that. No, I'm saying this is a human lung. It's being DNA tested, CAT scanned. I know the process. This is the pleura, this fabric bag that coats the lung. There's literally blood coming out of these, and I have some that actually the blood is coming out. I had 3 things DNA tested. This is our size of a human lung, but there was other ones that were giants, and I mean huge, and I'll show them to you in a second. All right, they have to do a thoughtful examination of evidence. I'm talking evidence. I'm not talking about theories and, flowery this and that. No, I'm talking physical. I'm a material scientist, and I do across the board. I'll talk physics, chemistry, biology, geology, space, meteorites, all of the workings of the body, the structure of the body, and I have to know all of that in order to present what I present, and I can present that, and I do know all of that, and I will stand up in front of anybody. Anybody. You get anybody. You get Derek Briggs from Yale to talk to me. Who won't? You get anybody from any of these schools. And I got so much to talk about. It gets so frustrating when I start speaking about this that I think I must take a short break. All right, so the better the school, supposedly the more they understand and the more they can convey to their students, so the students will learn more. Well, right now it's indoctrination. The students expect their trusted professors to have done their homework, which they haven't done. This would require thoughtful examination of the physical evidence regarding new discoveries, especially ones that dispute exactly what they're saying and may be directly opposite of what the professor is teaching. And it is. My stuff is totally opposite. That is institutional learning, but it is not happening. The story is very sad and long. And don't forget now, this goes back to 2015. And I presented all my evidence to Yale and Harvard and all of them back then. And these are the CAT scans of the large primates. All right, this is how this whole thing started with dinosaurs, basically. And this woman, Mary Schweitzer, who was a professor, and she was on 60 Minutes on TV, showing that she found soft tissues inside of dinosaur bones. Now, if there was soft tissues inside these bones, it would mean that they're not millions and millions and millions of years old. So they shut her right down. And they tried to destroy her. And they actually fired Mark Armitage, who was working with her. I don't know the details on it, but he was fired for making these claims, and he actually got a settlement because he sued. But they're still, this is 2017. She says, I don't care what they say about me. Paleontologist stares down and says, I know my work is good, and she's right. I had a little interaction with her. It didn't go all that well. It never does. But I spoke to her, and just for a second, and she just said, don't contact me again. I only work on my own stuff. But by that time, she was jaded, I can guarantee you, because she had been just so destroyed, absolutely destroyed. And she's still trying to eke out a living at doing this. In my way of thinking, maybe that's a wrong interpretation, but from what I was reading, she doesn't sound like she's real upbeat, and she's not getting any funding. She's not getting the kind of things that a good scientist would get, because she's not saying the things that a good scientist would say. All right, this is about Mary Schweitzer and how they really dismissed her and are keeping her pretty much suppressed. And it says no one except Schweitzer and her collaborators has been able to replicate their work. They won't look at the work. I have a DNA and CAT scans of specimens, and now they are understanding, and a whole batch of people are starting to jump on board with this, like Yale says, perfectly preserved, exceptional preservation, soft-bodied creatures. I just saw one came in. I forget where it is. It's somewhere around here. Anyway, hold on one second. All right, see, they're all starting to jump on board. Exceptional preservation, soft tissues in a new species of, however you pronounce that, Confucius Orononis. And then Yale did the same thing. They called it the Edakara biota. All that means is Edakara or whatever it was, is a place in Australia. And biota just means creatures. It's biological implications. This is from 2017, and this is all pretty much Nanjing University and field, pretty much, I think, China. And they're talking about preserving rare soft tissue traces of the ovary and wings. Now, when they say soft tissue, I think they're talking about it's in stone, but it was soft tissue, and they're showing what looks to me like it's in stone. But I have stuff that really is bloody. Now, it's not really soft, but it's, all right, you saw the one about Yale showing the soft tissue preservation. Here's another one, exceptional preservation soft tissue. When they say exceptional, it's the same as Yale saying. And it's the same, I call it fascia facilitated fossilization. The fascia surrounding all the organs and so forth creates a perfect medium for preservation in these wet, flat, silica-rich floods. This boiled out of a guy's body, and the reason it boiled out, because we almost got hit by Venus. You got to do some research and look into Velikovsky's research that he did on every single culture on the planet. Every single culture had the same story, and we almost got impacted by a fiery comet. And it boiled the oceans. It was so hot, and it disturbed the rotation of the earth, and everything swelled up from the bottoms of the ocean, salacious oozes, which is all this silicon. And it boiled the waters, and the creatures' bodies boiled, and the flesh boiled right off them. And it left these body parts. That's how you got the red bed, was the flesh. Then the gray clay came above it, which was the tendons and the bones and the other stuff. And then above that was the black cap, because that was the fallout of all the ashes and volcanoes and everything that were part of this catastrophe. So the thing almost hits the earth, causes the earth to go into gyrations, oceans go crazy, the atmosphere gets so compacted that virtually everything turns into water instead of hydrogen and oxygen. It combines because it's being compressed, just like in a compressor, and it turns to water. Only it would be boiling water, and it would come down on the earth. Secondarily, the sunlight, or the light from this comet, was 10 times brighter than the sun, they say, and it almost touched the earth. Just barely missed the earth. And at that time, all of this 3 layers happen, a Triassic, the red bed, the gray clay, the black cap. And Velikovsky recorded, and they say it's worldwide. It's not just one place on Earth. This is worldwide. And they call it the earliest fossilized creatures, multicellular organisms, the first ones that were ever alive. They said there was no bones or there's bones all over the place. This is a bone. They say, oh, no, that's a stone. No, it's not. It's a bone. And it turned into a stone. That's where the marrow was. This is, again, the fascia, collagens, it's phospholipids. They turn down into aluminum silicates. I know this stuff inside and out. I'll talk to anybody. I don't care who they are. Chemists, biologists, anatomists, I got it all. And there's no question that what I'm showing is correct. And I have the DNA and the CAT scans and everything else. And the ones that I showed, the first thing to Yale was they went crazy when I showed them the giants. And that was it. It was done. So, and they call it Edakara style preservation. No, it's Mudfossils. All right, so, and it worldwide, see? Worldwide, in this one strata, which is the red bed, the gray clay, and the black cap. Worldwide, and that's exactly what I have here. And they're human beings, and some of them are giant. All right, let me read you this. This is from 11 of 2015, Fascia Facilitated Fossilization. Now, this is just sums it right up. This was 8 years ago. Fascia is instrumental in perfectly preserved fossilization in wet, fine, continuously wet mud fossils. The product can be exact copies of the living creatures, flesh, colors, and all. Yes, just like that. So that's basically a piece of meat. And that's the fascia that surrounds it, just like it does on a piece of meat. Now, and I have all this stuff extremely well documented. Now, all fascia is made of collagens. All right, collagens are this rubbery stuff, and they're made of collagens and keragens. And this is the fabric. It creates the fibers and elastics, so they can move around. Like your lung has to do this. Your lung has to, and that's what this bag does. That's what this rubber bag does around this, and it's all got all these little fibers in there, so that it can stretch and come back to where it's supposed to be. All right, so all fascia is made of collagens and keragens. That's the fabric that creates the fibers and the elastic sheets and wraps everything individually in your body. Everything is individually wrapped. You know, your organs, your muscles, your tendons, your bones, that's all covered with fascia. Everything has, it's a membrane, basically. Think of it that way. Separates one thing from another. Now, they all come together in one giant system, which is, they now call it the interstitium. They just announced this in 2018. I was talking about this in 2015. Now listen to this. There's over 20 types of collagens. I think there's 28. And there's keragens as well. They're different ones, and they work in different areas. Some protect you from acids, others from caustic areas, others mineralize for strength and give you bones and so forth. And the variety is amazing. The fibers are infused with silicon. All right? That's what I found. They were infused with silicon. Well, guess what Yale says? Exceptional preservation promoted by silica, which is silicon-rich oceans. Same thing. All right, let's just continue on here. Mud fossils are exact copies of original creatures separated on the fascia and skin separation borders on their planes. The fascia separates every cell and structure from its neighbors. It's got a membrane when you're alive and when you're dead. In this particular situation, fascia protects every bit of you from invasion, including a complete fabric bag that is your skin. Your skin is not just one fabric either. You have tough skin and fine, thin, all different types of skin, and grip skin on your fingers, which on your hands. I'll show you that in a minute. All fascia is made of collagens and keragens, and that's where I went back into that. Skin has 50 times the silicon as other tissue. That's that silicon-rich stuff. It's exactly what everybody's finding now. But they wouldn't even look at it. Now all this stuff is gone. I had to take all this stuff down, so any links in here they're not going to work. But I did work with people overseas with fascia in primarily Germany. But they were doing. Nobody understood fascia. When I first started with fascia, about 5 years before this, because I was saying, how does this happen? Why is this happening? It was all these organs, and like even this goose, the feathers are nothing more than fascia too. They're collagens, and everything I had was separated by a membrane, the membranes. And I said, well, how did this membrane do what it's doing? Well, membranes are made of phospholipids. Phospho is phosphorus. Phospholipids, phosphorus and fats. When they boil down to aluminum silicates. Okay, this is bringing back a lot of memories. This is from the same paper back in 2015. Fascia was thought to be mere packing material and virtually inert. It was just like a fuzzy. They called it fuzz. It has long been ignored as a subject of research. Recently, however, there's been new equipment that allows viewing of the fascia as it does its job. I was watching these experiments. Anyway, once again, as in many experiments, they were totally unexpected results. Fascia is a tubular, fluid-filled fabric network. Listen to that. Fluid-filled fabric network. And I said it instantly reconfigures and so forth. And it could even be fiber optic wiring, but I don't think so. I think it's all related now to the lymph nodes, lymph system. But it is a fluid-filled fabric network. All right, that's where the fascia, this, they call it interstitium now. It's right below the fascia, or actually right above the fascia, because it protects from invasion. So you'd have that fluid-filled highway just above the membrane. Well, actually, yeah, all right. So the interstitium is this white fluid-filled layer right here. By the time they had him in the autopsy room, it was flat. They just thought it was a piece of flat sheet. And it's not. It's a fluid-filled highway. So it sits on top of the membrane, and then the membrane protects this, which is your vital flesh, from attack from above. This stuff sort of can fluff off, and this is no big deal. Once you get through this, then you're in trouble. That's when you get a serious, infection. And then, cancer is an invasion where something grows through there, and it grows into this tissue, and it makes a tumor. Otherwise, it's just an invasive gash through this protective barrier. All right, now, so this is the interstitium. Now, this is 2018. Meet your interstitium, a newfound organ. Now, the guy that claimed to have found this and made this paper in 2018, they won't publish his work either. I don't know, somebody published this, I don't know who did, but it's not published in any of the journals. And, well, hold on a second. All right, so this is New York University School of Medicine pathologist uncovers a potential new organ, setting off a fiery debate. Now, this Neil Theis doctor found that the interstitium, long thought to be mere connective tissue, serves a unique and critical function. This is the interstitium, the fluid-filled highway. And I show this in my paper. I know I presented that to you, that they thought it was just merely fuzz for forever. But it's the fluid-filled highway. Anyway, I know I showed you that. Hold on. All right, so this is the doctor that's put out this paper. And he published a study in the Scientific Reports promoting unusual network of fluid-filled compartments under the skin. That's the interstitium. That's what I was talking about in my paper. Collectively known as interstitium. Is it a full-fledged human organ? Yes, it covers the entire body, and it all focuses into one giant system, as I reported in my paper in 2015, by the way. Now, listen to this. The report was greeted by 2,400 news articles, 3.8 billion online views, and no small bit of controversy. Scientists continue to debate the implications. Now, this is 2018. He put out his paper. So that's 6 years ago. Now, is it just connective tissue, or is it this interstitium? It's a fluid-filled highway, and I know exactly what it does. It carries the enzymes and bacteria through your body to attack invaders. It's very, very, very important. I've done a lot of research on this since then, because I already knew about this long before he did. They thought it was just a stiff, flat plate, but it wasn't. Hold on a second. Let me come to something important. Hold on. All right, listen to this. This is typical. This is just totally typical. And he's in the scientific community. I come with my stuff. They don't. It's just there's nobody who looks at it online. He had billions of hits, and he's still. Listen to what he has to say. Okay, you can never tell where some discovery is going to come from. He says, what has surprised you most about the scientific community's response? So Neil Tice says, I knew there would be pushback and criticism because that comes with anything that changes paradigms. Boy, you're not lying. We're an interdisciplinary research team, so far including endoscopists, a hepatologist who is also a cell and molecular biologist, a pathologist. We came in from left field, and naturally those who are in the fascia world or the interstitial world say, who are you to say we missed all of this stuff? I would have the same resistance if someone from neuropathology made a liver discovery that upended my assumptions. So he's in the same boat I'm in, but only he's part of the establishment, and they won't listen to him. Now, despite the study's significance, scientific publishers weren't breaking down your lab door to publish it, were they? And he says, we sent it to all the top biomedical journals. None of them even reviewed it, because they didn't think it would be of interest to their audience. It turns out they were wrong. Every time we got a rejection letter, I thought, we're on to something so big they can't see it. No kidding, Neil. How have you handled the massive response? The media explosion kept me very busy. I experienced 4 days of fame, and now it's over. That's that's a relief. The more important thing is that we suddenly have these relationships with physicians and scientists around the world who are picking up on this new understanding of the interstitium and running with it. No, they're not. They're not, Neil. They didn't pick up on it, and they didn't run with it. We have to adjust to the idea that we're not going to be able to do all the work on this project, and pretty soon we won't even be able to keep up. The baby is up and walking. No, the baby just fell down and it's not walking anymore. Could the interstitium lead to new categories of disease diagnosis? Absolutely it can, but they're not looking at it in the right way. I'm telling you, they're not. All right, and he says, you made this finding with technology allows you to observe living cells instead of studying dead ones. Does this approach have broader implications to other parts of the body? Well, yeah, it's your whole body. Every single organ in your body is protected by a layer of interstitium, which is that fluid-filled highway. So what happens? You try to get invaded, that fluid-filled highway sends all kinds of enzymes and bacteria to stop that invasion. Now, you need the enzymes and bacteria living there, so you can't damage that microbiome. Once that microbiome is damaged and you don't have the bacteria, you can't create the enzymes. You can't create the enzymes, you can't create the chemistry. Enzymes are the things that do the job. They work instantly. They do more work in less than 1 second than would happen in years if you didn't have that catalyst. And that catalyst is an enzyme, and that enzyme comes from a bacteria. So this is your protective zone. You can also be invaded through that zone too by bacteria, so it goes both ways. But primarily, if you have good bacteria, it's going to kill the bad bacteria, because it's a fluid-filled highway. If you need more good bacteria, the more will come. But you have to have it living in your body, or none will come. And it lives in this layer. Now, this is a lung. So what's inside of here? Blood. Well, what is blood? It's a fluid that has sort of a certain pH and it has a certain chemistry, and this will have to protect that certain chemistry and that certain pH and all that. Now, if that was a kidney, it would be different. It would have a certain salty or of a liver or gallbladder or pancreas, or all those different things have different coatings, different membranes. That's why you have all different types of cancer. Because the membrane, that particular membrane, is not doing its job. It's being invaded. Basically, that's the way I see that type of stuff. And research points it out pretty well. There's like hundreds of different cancers. And every one of them, like bone cancer, liver cancer, every one of the organs has its own membranes around it. And then, anyway, it gets a little deep, but nobody would even publish his stuff. And he's a doctor. And he's putting this stuff out. No, they don't want to know anything about it. They're not knocking down your lab door, are they? Well, I can tell you what, they nailed my lab door slammed shut years ago. Okay, my friends, this is going to be exciting. This is a brand new body part. They don't realize that is a separate part. It's not part of the fascia. It's this little flap right here. This is in a living one. This is in a mud fossil. I discovered this body part, and I also discovered the interstitium years ago, looking into mud fossils which preserve in exceptional ways. So let's get started, because this is kind of a long and tangled story. All right, this was from Helix Biolabs. They were absolutely fabulous, very, very professional, and I know they took heat for doing this. But I did talk to the owner there, and he said they will stand behind the result, but they want you to understand I was the one that extracted samples from these mud fossils, sent them the samples that I extracted. They did not extract it. I extracted it and sent them the samples. Then they did the PCR and the DNA extraction from these products that I sent them. And this was a 36-inch tip, which I'll show you, the lung, which is this here, and a mud tip, which I'm going to show you. I have a CAT scans of that. Now, this gets pretty detailed. I'm just going to go down to where it talks about all kinds of stuff that's, a little over my head. I don't care about it, just so long as the sequences were correct, and they were, for mitochondrial human DNA. And right here it says excellent quality DNA from the 36-inch tip and the lung. All right, excellent quality. And here's what it was: Homo sapien mitochondrial, B gene, and D loop region, both of them. The lung and that's modern DNA, human. All right, and these were all the CTAG sequences. This was no puppy test. This was expensive. It took months to do. They had to do all kinds of elutions and so forth. All right, so this was serious stuff. And here they are right here, Homo sapien. Now, so there's Exhibit A. All right, this is Exhibit B from a Fascia facilitated fossilization up on academia.edu. And I was told that you can't get into this anymore. I can, but apparently they stopped. For some reason, nobody else can seem to get into this. But it's up on, I had like 45 papers, something like that, up on academia.edu, just so that I could get them somewhere so that it'd be locked down in stone that this is what I presented. And this was about all of the fascia. You're going to see this quite clearly in a minute. All right, check this out. This is the head of a bone. All right, it's a bone. Now, the outside is covered with fascia. This is where a ligament attached right here, which attached it to another bone. They were attached together. But there's other things they call bone foramen, where blood vessels flow from the tissue outside of it into the bone. Now, this is one of those cases. All right, that is a scab. That's a scab, and I can't remember if it came off of this bone or not, but they all have them. They all have bone foramen, and when they come out of the ground, the blood leaks right out of them, and that's blood was inside there, leaked right out. And not only can I show you that that's a scab, you say, oh no, that's just red iron oxide. Well, yes, but guess what's underneath it? I can show you what's underneath it. Is right there is what they call fibrin. All right, this is clotting fiber. All right, this is what it is between your scab and your wound. All right, now, how did this blood get out of here? Where'd that come from? Didn't it maybe just puddled in there? No, it came out of these spots right here, as I will show you right now. Those are the two spots right here. That one is the black, you see it? That's where the vein blood came out, right out of there. This one here is the artery, the red blood. You always have a vein and an artery. You have to. One brings the blood in, the artery, and the vein brings the blood back. That's the system. So here's the end product with everything stripped off, most of it. And here's where the clotting fiber is still there, and I push the scab to the side. But these are where the black and the red is down here. And here's before it was molested at all. It just came out of the ground. Now, I have other ones, lungs, that they just actually, blood actually bubbled out of it. When this first came out of the ground, this was really leaking blood out of here. It's red right now, but it was, when it first came out of the ground, it literally leaked. And I drilled a little bit deep in here somewhere. And you know, it's, I think maybe right in there. But anyway, all you need is a little pin drill, and you just get a little bit of dust. That's all. You don't need much to test this. It's not like a big chunk or anything. So anyway, this goes back 10 years almost that I had these DNA tests done. You can get, I'm sure you get DNA out of that. And these were excellent quality. They were in very good shape. I'll show you this in the microscope. When I put a little water on there like it was when it first came out of the ground, you'll see. Okay. Everybody says, how do you get blood out of a rock? Well, I'm going to show you. It's very, very simple. First of all, you have to have the right rock. Now, that's a lung, which is this lung right here. And that red bloody spot is right here. Now, I put a bunch of water on there, and it rehydrates very fast. It turns back right into blood. Now, this one preserved in a certain manner where it turned into all basically silicon. All of these little circles are alveoli, which are these things in your lung, which the air goes in. Anyway, this one, all of the pleura stripped away, so you can see the alveoli. This one has all of the pleura on top of it, which is the fabric, which is the one we're going to be looking at in the microscope here, uno momento. Now, what am I going to show you? I am going to show you this spot right here. Right down here. Now, I think I drilled a hole right in there. It was somewhere down in here. And I got, first of all, you got to clean it very good. All right, let's go through the process of extracting DNA. You have to clean the area extremely well. All right. Then you have to, of course, be careful with gloves and all that business and mask. You drill into it. You don't have to drill a big hole. You just drill enough deep in there where you're past any possible contamination. Then you drill a little deeper, and that is what you extract and put it in a little plastic bag and label it whatever it is, and off it goes. Now, I'm going to show you what this looks like in the microscope right now. All right, that's the picture I took years ago. And up here is right now in the microscope. Now, what you're looking at is it's wet. All right, it's wet. I'm going to scratch around in here, and you see what happens. It turns right back into blood. You see it? You just got to get a little touch below the surface, and it's just nothing but blood. That's what a lung is. It's just a big bloody mess. You see that? Look. You get DNA out of that. I guarantee you that. There's no question whatsoever. And what you got to do, you get in below this surface. But he said it was just like, it was like raw blood. I mean, you can see it. You can't see that. I don't know how you're missing it. Okay, so that was Exhibit B, blood from a stone, CAT scan, DNA tested, human mitochondrial DNA. And it can be retested. It's not, I don't mind. Somebody can retest it. That's fine. I'm not going to do it again. All right, I just showed you the lungs. I showed you the scab in the bone foramens, and the blood vessels. Now, what freaked them out was the giants. And what giants, Roger? Well, these giants right here. This is it's over 3 feet wide, a hand. Now, this is a left hand. This is where the little finger goes down this way. Do your own hand. You're going to see there's a little tendon that runs right down here. All right. This is, of course, the bumper in your pads of your hand. That's the cleave in between where they pinch together there. This is what they call grip skin. And you say, well, what is grip skin? Well, grip skin is, it's a dense, coarse layer that's on your fingertips and the palms of your hands and on your, I believe it's also, you call the same sort of stuff on your elbows. It's a real coarse skin. It's on your toes and on your soles of your feet. And it's made to basically wear off. And you see how it's actually peeling off here? See it peeling off? It's like silver looking. That's because underneath you got a lot of flesh, and this protects that flesh. And you got a lot of blood in your hands, a lot of blood. You see how much blood you got in your hands? That's the terminals. They come down the artery and they come back to the vein. And every one of them has the same thing. They have these little tips, 2 on the side, 2 on the end, and 2 on the other side. Vein side, artery side, that type of thing. Now, I said I had CAT scans. Yes. Here's one of the fingertips. Let me go get it. Hold on. All right. This is the fingertip that is here. I'm going to show you this in a second. But this is the actual fingertip. Now, I broke this piece off, and we can see the bone underneath, because this is where the bone meets up right where this tendon is. The bone is offset, and I'll explain to you why it's offset, because most of your bones are straight in the middle of your fingers. I'll explain to you why it's offset. You see the bone is right here. There's a tendon here, and the bone is here. And that's the artery, vein. You see the black, see that hole there? See right there? That's the tendon. And that is the vein. This, let me put this down. All right, let's back out of here so we get a perspective. Tendon, vein, tendon, artery. Arteries just run out. The blood just runs out of them. The veins, they plug up and they turn black. Now, this is the bone. You see here? Now, this is what they call a 2D CAT scan. So they take it from every single angle and they put it together so that you get the best look at it, and it really works very, very well. This is from Jesse Garant and Associates. They have 2 places. They are the tops, and basically, I think they might be the best in the world. And they did 7 CAT scans for me, no charge. No charge. 7 of them, because they are scientists. I don't think it did anything for them, but I'm hoping that somebody understands these are real scientists. Now, this was the 2D of this fingertip. Now, they also did 3D. I'll show you that. Okay, this is like a real mud fossil. This has been completely transitioned into what would you call mud stone. However, you can see that shadow right there. You see, actually you can see one here and one right there. 2 different rings. All right, can you see that? Maybe you can, maybe you can't, but they're there now. And you can see there's blood supplies and so forth, but you really can't make out a lot of detail. There's just not a lot of detail to make out in there, because it's what's called nucleophilic invasion and substitution. What was here has now been removed. There's a leaving group and an invading group. So the invading group is mud, and it took over everything that it could. Now, this piece right here is broken off, and I can see the bone, and I will try to remember to show you that in a microscope, because I got the microscope fired up. We might as well do a few shots. But this is the problem with the 3D. as you pan through it, you don't see a whole lot of difference. 2D is excellent. Let me show you another one that actually shows, you see here, looking at the top of that, it looks like it could be a fingertip. It could be a fingertip. And where's the fingernail? There should be a fingernail if there was a fingertip. Well, it's eroded off, but it leaves signatures. And what signature would that be, Roger? Well, let's see. And there it is right there. There's the fingertip. Well, this is the fingertip, but that was where the fingernail was, the bed, you see it? That's the beauty of the 2D. You'd never see that. This is the same fingertip. And it's there, but you really can't see it. All right, you see that gash right there? That's this gash right here. You see that right here? This is where the tendons lock in. All you got to be is an anatomist to look, and that's where the tendons lock right in. All right, so you got that one. You got the end shot. These are the best ones to see. The two, I mean, the anatomists tell me, yes, this is true, but don't use my name. I don't want to be involved. One guy said, what are you trying to do, ruin my life? I said, you know, you got to, like, I paid for these DNA tests, so I don't think he was happy about me talking about him, but he did a fabulous job. It's just the problem is in academia. It's not him. And this is all true stuff. So I'm telling you right now, you're going to see we're in a bad situation when it comes to academia. They rule, and you can't speak up. See what you think. Make comments. Tell me if I'm wrong. Remember I showed you the grip skin on that big hand there? Well, this is a fingertip. This fingertip is 3 feet long. 3 feet long. This is the pad that goes between bones so that they rock on that little pad. This is literally the fingernail. And it's in good shape. Now, when I first found this, the surface gets, you can't get into any blood on the surface. You got to get into the surface, and I didn't want to bring a drill out there or anything, so I just figured I'd just smash the thing home, which I did. And I just smashed here, and a piece popped right off, just like a scab. And it was literally the skin, the grip skin, popped right off, and it still has the fingerprints. And I'll show you that in a second. And it wrapped right around there where the fingernail was. And underneath is all the blood. I took it out, and excellent quality DNA was in there, because fingers and so forth are saturated with blood. Saturated. All right, now this is a standard standard finger, because the bone is right in the center. Remember I showed you this one, the bone is way off to the side? That, my friends, is a thumb. It's a thumb from the right hand. You see here? You see that lump sticking off the side? That's that lump. The bone goes up this side. The lump sticks off to the side. You see it? So that's a right hand thumb. And these are the tendons. I mean, there's nobody can deny this. It's undeniable. And the DNA tests and the CAT scans and all of this stuff. And look at this. Remember I showed you, where was it? The giant finger, this one here. The piece I broke off still has the fingerprints on it. And the blood was excellent quality. Here's the fingerprints right here. Look. This is what fingerprints look like. They have these little sweat pores in them. See them? And you have this big pad just comes right off. That's it right here. Here's the fingerprints. And the fingernail was right up here. My thumb is about the same width as one of these ridges in the fingerprint. This thing was huge. This is giant. This is human. Now, this popped right off just like this. It's almost the same. It just like ripped right off. And right below is just full of blood. And I mean, it wasn't red flowing blood, but boy, I'll tell you, not too bad. And it was excellent quality, so Let that speak for itself. I'm in the Triassic rock area right down the street from Dinosaur State Park. This is red bed. There was gray clay on top, but some human footprint stepped through that gray clay and it pushed down through the red bed. So that means they were both wet at the same time. And then this was on top of this, which is the black cap. And that's all the fallout from all of the catastrophe that happened. And I even have a foot that seems to fit pretty damn close into there. There it is right there. And that is where the bone comes down, like we have a bone that comes down and rocks in the cradle the same way as that. It's called the tibia. All right, and then we have a fibula that comes down the side, and between the two of them you can do a bunch of stuff with your foot and move it around. The same thing as this. The same thing. Now the only thing is, these don't have toes. This is the foot itself. This is not a shoe. And apparently, I think it must bend right here, bending like that. And I believe this is where the springs are inside here. Right, because they have springs. We have other ones that actually show the springs very clearly. And I believe that's a pin where those springs rock. This is engineered stuff. This was pre-flood, pre-great dying. This was Triassic stuff. This is supposed to be before there was even any bones or teeth or anything. No, there was all of that stuff. And it looked like somebody was playing around with it. And even Yale said it might have been a failed evolutionary experiment. And I agree with that. In their article, they say it could have been a failed evolutionary experiment. Because almost everything on Earth died under this gigantic flood. All right, so stay tuned. Okay, I'm hoping my microscope holds out here. It crashed out from me, so I might be out of sequence here, but it is what it is. This is that big fingertip. It's this one right here. All right, now I have it in a microscope. The microscope's kind of high up here. And we're going to be looking at this piece right here. This is broken off. So we're going to see the actual bone right in here, I believe. Let's take a look. All right, this is just too crazy. This is the piece that was broken off, and this is literally the tendon. So I'm going to remove the tendon. And right below it was where the blood came in, the black blood. All right, this is literally the tendon right here. Whoops, hold on, let me move this out of the way. You see that spot with the real orange looking stuff? That was where the tendon came in, right there, right exactly there. And it comes down this way. This is tendinous stuff, and it's fibrous. And then it broke off here. Now the blood ran with it. This is where the blood is. You see the color of the blood? So that's the piece that broke off of this other piece I just showed you a minute ago. Here it is in its totality, and that's the piece broken off. And that's the blood running down at the bottom. And you see this color right here, this orangey looking color? That's where tendons lock in. You see over here, same thing here. These are all tendons that lock in right here to that fingertip so that it anchors the tendons in. And I believe that's sulfur, but I'm not sure of that. So anyway, that's kind of interesting. All right, let's see what happens. I'm going to put some water on there. It's hard for somebody that doesn't know what they're looking at to understand what you see. I got to be honest with you. I'm sure you're going to have a hard time understanding what you see. However, I will try to make it as clear as possible. Now, water sometimes gives it a little bit better of a look, but sometimes it just too much flash now. Well, this is the bone. I'm going to have to get closer to it for you to see the bone. But that's the little bumpy looking bony stuff. And that's how this flesh locks into that bone. Remember, I broke this piece off. That's all bone down there. And this came off the tendon. And that's where the tendon ended up locking in. Right up in here. Now let me see if I can get a clearer shot of this. I know this is all wobbly and nasty. All right, that's probably not too bad. Now I'm going to have to play around with the light a little bit. And focus a little bit. But this is the top, where the top of the flesh is. This goes down till it meets the blood, and right over here is the bone. I'm going to come right down on that bone. You see that? That's bone. And that's in this giant fingertip. See, there's bone, and there's some blood right there. But all this white looking stuff here is bone. And that's how things lock in so they don't get all ripped and torn apart. And then this, of course, is the flesh out here. Well, it's not, everything's eroded. But it's biology, trust me. Whenever you see these little line-looking things, that's some kind of tendon-looking stuff. And that's where the bone was. I think this might even be a better shot of the bone. Hold on. Well, it's bone. Just trust me. It's bone. And then you get over in here, and there's where your blood meets up with it. Remember I said the blood flows right along with that tendon? There it is right there. So this is biology. Nobody can explain this. In the middle of a fingertip that's been DNA tested and CAT scanned. I mean, this is not right that it should be just dismissed. And when they keep saying, oh, nobody can prove this, nobody has any evidence, if you won't look, obviously there's no evidence when somebody won't look. All right, watch this. This is that tendon area right there. Now it's a little shaky because I don't have it locked down. But right there is where the tendon enters. I'm going to remove this because this up here is the bone. That's the bone right there. Now the tendon came down below, down to here. Now let me see if I can stabilize this. Hold on. Alright, now check this out. I'm not going to focus in, but right here, I'm just going to remove this, and look what's underneath is where that went to, which is right there. You have to take time and focus in on something. Now, take your time and look at it. I got to lock this in solid. And we'll focus right down in there and take a real close look at that tendon in place. Because this was right on top of it. And that's right there is where the tendon locked in and ran up. And I'm going to put some water on it. It should show up a lot better. But this is the separation between the tendon and where the bone is way over here and the blood, actually, because the blood runs right along with the tendon. All right, I put a little water on there. You see how it brings out the colors? This is all blood here. That's vein blood, it looks like to me. The black is normally vein. Now, this right here is the tendon, and it's running this way. All right, these are the fibers, they're broken off. Now, let me dry that up a little bit, make it a little easier to see. And then we're going to come right down on top of it. You see it? Still kind of shiny. We're quite a ways off. But you can see the differentiation between this tissue, that blood, and this tendon. Now, we're going to come right down right on top of it. You know, close enough so we can get a pretty good shot. There's the center core. That dot right there, that's the center core of the tendon. And the whole thing basically was right in this circlish area. Hold on. Basically, the tendon ran right up straight through there. And right here and here, there's the core. It's pretty obvious right there. That's exactly the core right there, that circle. You see it? And that whole area, though, was tendon. So you had the tendon, and then you had around the tendon, you had some kind of stuff. Same stuff over here as over here. And there's a tendon in the center, and when it ripped out the whole thing. And all these colors are colors of skin and blood. That's just what it is. All right, I'm going to try to explain this, but this is coming down from the top of the flesh down to where it meets with the blood, and then over here where the bone is. You see all that white stuff, that's bone. But there's also flesh attached into it. You see all the little fleshy spots holding itself in. This is pretty fresh stuff. Once you break down inside, you get into the bloody areas, and these are little blood spots inside. Let me turn the light down a little bit, see if that makes any difference. Oops. All right, you see the red blood and the black blood? That's it. And the rest is bone. But that's how it locks in to all those little tiny crevices on the bone. This is not just a rock. And it's been DNA tested, CAT scan, everything else. So it's just nonsense to like I say, it's very, very frustrating. This was all done in 2015, 100% done. All right, I think it's sufficient evidence to say, to support some kind of an investigation into this to see if it holds any validity, which it obviously does. I mean, I've done my homework, and this is 10 years ago, more than that, but you know, just locked down solid evidence. 9 or 10 years ago, no way to dispute it. So, not understanding is one thing, but when you know the truth, and I think anybody in there, you know, there's no reasonable doubt here. Let's put it that way. Where everybody's talking about trials now, there's no reasonable doubt here. So I put my exhibits on. Let's see the defense, and it may end up that way too. This is serious. This is serious neglect, breach of what you would consider to be moral and ethical and legal obligations. And if you speak up, they fail you. That's hard to believe how they've got this system working for themselves. You know, you got to pay them, and then you got to say what they tell you to say, and remember what they tell you to say, and then go home and say that, and then they'll say you're smart. If you don't, they say you're stupid, and keep your money. Okay, this is outrageous. This, I'm claiming, is a new body part. All right, and it's not just in human beings. It's in every creature where one set of, like in this case, it's muscle meeting what I believe is tendon. And you have to bring that fascia into it. And then you also have to transfer the fluids that are in the fascia. And this is a rock. All right, and it's a Mudfossil. But you see right down here at the very end, you won't be able to see this. I'll put it in a microscope. There's two little dots at the very end, and that transfers the fluids into the next interstitium. And they're going to be different types of interstitium, because one is going to protect the muscle, one is going to protect the gallbladder, but they're going to have to connect into one giant system. That's where invasion happens in different types of membranes, because membranes are made of different chemistry. I don't care what you say. Now I'm going to show you something that again is a piece of I call it a Spurlock, and it's attached to this fascia, and it's got all this little gnarly stuff up back here, and then it comes out here and would have been laying into something else, another body part, whatever, across your shoulders or whatever. But this is the attachment part, just as this one is up here. All right, whatever this is, I don't know, but it's attaching to here, and I think that's like a shoulder or something. This is a sports injury. So it's wherever this was attaching to, it's ripped away. Let's go with that. Now, I have a good friend who is an anatomist. He's an autopsy anatomist, and he teaches all over the world. And I showed him this. I've been working with him on and off, asking him questions about anatomy for 15 years. And he's the one that I first originally started talking about the interstitium with. What they called it fuzz. I said, well, what's the stuff that's all around these organs and stuff? And he said, oh, that's just fascia, that's fuzz. And I said, there's something else going on. And then we kept going on with it. And he did actually do a video, say, reconsidering the fuzz. And he's a brilliant guy. Now, I asked him about this. I said, this little latch here is the same as that latch. What do you call this? He says, oh, that's just part of the fascia. And I said, well, I believe it's a whole different thing than the actual fascia. It's like a different little organ. And I'm going to show you why I can say that. And he said, well, if it is, nobody knows about it because it's considered part of the fascia. And this is a guy he's one of the people in the world who would know. He's one of the top people in the world. All right, these are lungs. That's a DNA-tested human lung. No question about it. And it has that little flappy thing. Now look at this. This is not a human lung, but it's a lung of some sort. I'm going to put it in a microscope in a second. That right there is that latch. I'm going to put it in a microscope, you'll see it. That's that latch. Everything else is eroded away. This is nothing eroded. All right, but there's that latch right here. All right, but it didn't erode enough to show up as its erodedness. All right, this one did enough to you can see it quite easily. It's the same latch as this, only this is on a lung. That's on a piece of meat. And this is definitely a lung. I'm going to put it in a microscope and you'll see. And I'm just calling this the Spurlock right there. And it is not the same as the fascia that coats the lung, the pleura. It has nothing to do with that. Totally different substance. All right, that's pretty obvious what you're looking at there is that little flap. And this is the bottom of that lung. These are where all the alveoli were. And they fill in with quartz. In this particular case, sometimes they could completely evacuate, and sometimes there's still blood left in them. I have them in all those different conditions. And then I have them with the pleura completely on it, like I showed you the one I had DNA tested. I have it with the pleura completely gone and every bit of it turned into quartz. and I have them with half pleura on and half pleura off. I'll show you those right now. And then let me show you all of them. All right, you saw that. And that was from right there. This little one right here. All right, now, this was the one I had DNA tested. All right. And that had that same flap too. It just hasn't eroded enough to where it sticks out by itself. And this still has all its pleura coating all that. Now, here's a lung from, I don't know what it's from. I don't think it's human. Could be. I don't know. And it has that gnarly looking thing too. And this didn't erode the way this one did. This one still has a lot of its pleura on it as well. All over this thing. This one eroded away, so it just left that tab, whatever it is. It's inside all of these. They all have it. And hold on a second. You see this one here? The one I showed you here has all of its pleura on it, 100%, just like it came out of the box. All right. This is the one. There was DNA tested, CAT scan. I tell you 100 times. I tell you that. That's all. The pleura is actually flawlessly still on there. The blood's sucking all everywhere, and it's flat as a pancake. Now, that's all the pleura. Now, this one has the pleura has eroded and pushed away. It got squished and moved away somehow. So now here you have this, which is, in this case, it's down here. Now this is the pleura. In this case, it's all out here. So I have some of this and some of this in the same thing. And again, flat as a pancake on the back because it was in a great flood. I'm in the exact spot where the Triassic rock is. All right, and this is all Triassic. This is all mud fossils. These transition due to nucleophilic invasion substitution. I've said it a million times. I've done it. I've made it myself. It's not hard to do. It doesn't take long. I had it around here somewhere. I don't know what happened to it, to be honest with you. But it turned into, well, I have pictures of it, but it turned into a leathery substance, which upon drying out would turn into stone. All right, but it was somewhat flexible. Anyway, this is not going to be hard to do. If somebody wants to recreate this process, I know exactly how to do it. And it was due to silica-rich oceans. You need silica, and you need to boil the creature in its entirety with the blood and all of the tissue and everything, the hair and all that, in a basin of some sort, which this is what I am. I'm in the basin, which means it's just boiled in this area. They boiled out, and all the body parts just boiled away out. So you need to do that, and for a very, very long time. At probably, I'm not sure what the temperature would be. That I don't know, but I do know that you need a lot of temperature, and you need a lot of silicates, and you need a lot of salts, because there will be ocean water everywhere. And then you need to boil them for a pretty good time. You know, a long time. I'm talking at least a week or so. And it depends. Some of them boiled right completely out. You see, this must have been in a real hot area, and just boiled completely into nothing. All of those were alveoli in the lung, and it's completely gone. So to my way of thinking, there was a lot of acids or salts or a lot of heat. But something created that, and something created this. This is like transplantable. This thing is just almost just like it came out of the box yesterday. It's perfect. But they all died in the flood. That's my, and this one here, the same thing, flat. They're flat on one side and basically perfect on the other. Flat on one side, and somehow this got eroded away and pushed off and ended up with the lung tissue right there. That's where all the alveoli were. This Mudfossils changes everything. Nothing is the same today. Nothing. And I haven't shown this ever yet, but I should. This is the guy's lung. That's the top of his chest. And his whole body is here all the way to his back. There's some little bits and pieces of bone here. And here's going down through his lung, all of the alveoli in his lung. And this is all his backbone stuff. And that's, I got to dig into this a little deeper, but that's the bones. There's a couple little bone pieces. So he must have died like this. Basically, along everything would have been here, and the water ran off there. You can see where all the water ran across it. And these are the alveoli of the lung. The lungs preserve. I mean, anything with blood in it, hearts, lungs, livers, fabulous preservation. All right, I know this has run very long, but this is the, I'm going to just finish it up with this. This is the lung that has part of the pleura detached, and the lung is just sticking right there. Now, I put a little water on there so we can take a quick look at it. Hold on. I put it in this little clamp here so it doesn't get jiggling around too much. All right, now, here we go. Now, I'm pointing so that half up here and some down here are going to be in the same shot up here. And I got to turn the lights off so we can see it. Hold on. And I'll have to focus it in. All right, now, let's start way at the top. You see up here, this is the actual pleura. This is the part that didn't come off. Now I'm going to put a little water on there. Water always brings out the chemistry better. Now, you can see that's what the pleura of a lung looks like. If you know what they look like, that's what they look like. Now, going down here to the alveoli, that's the alveoli down there. Let's see if we can get down there. So we're moving down here away from the top down. And we're coming closer and closer. And if you can see, there's a, you know, I mean, it's just bloody blobs. They're tiny, tiny little alveoli. I thought they were much bigger than this. But they appear to be extremely tiny. And that's what I did find in that other lung that I showed you the little latch on. The little tiny balls in there looked like, at first I thought, nah, they can't be. But they are. They're just tiny little things. Those are the alveoli. And that's what transfer the oxygen into the bloodstream. Hold on. Oh, wow. Look at this. This is the first time I've seen this. These are the alveoli. You see it? There's one here. There's one here. And all of that blood mixes through those little air holes. Those would have been air holes at one time. You see how round they are? This is the best one right here. And you can actually see the holes there. Maybe I can even get a little closer. No, I cannot. That's it. But anyway, you saw that. Those are the alveoli. And that is in that little lung here. They're very, very tiny. I thought they were much bigger than they are. But that does account for these little tiny, the one I showed you here, all these little tiny balls, those are alveoli. I thought, boy, those are too small to be alveoli, but no, they're not. Even in this, they're basically about the same size. They're not much bigger. Yeah, these things are tiny. These little things here, they're everywhere. But they're just tiny, and all that other stuff is blood working its way around through all those little alveoli holes. You see them? This is all blood. These are the colors of blood, and all those little holes are in this particular lung. Look at this. This is that little bitty tiny lung. The same alveoli. They're little bitty round balls. I thought they were much bigger than this. And they're all over the place. And this is the one that had a latch on it, which is right there. And it's got some really gnarly stuff. Hold on. Right up in this area here that latches this into that organ. That stuff right there. Right up in here, some really gnarly stuff. So that's where it transitions from the fascia into this thing here, which I call the Spurlock. And right at the tip, I believe these are the two spots that are where it transfers the interstitial fluids to the next organ or wherever it's investing into. And I'll show you the one on my other one right now, and then that's it. I'm going to lock it up for today. I know we've gone very long. There's a lot to go over, a lot. All right, that is a hair follicle. It's 1,000 times bigger than a human hair follicle. That is where the erector pili muscle attaches right there. That makes your hair stand up on end. That muscle goes right to there. There's the core of the muscle. All right. On the other side is the sebaceous gland. You see that right there? That's this. Now it was up further, but it's just slid down. You see it all flopping around the top? Now, the hair follicle comes down through the bottom, and the root starts way down here. Now the hair, vein and artery, blue and red means vein and artery, are right there, the two dots. You see it? This is extremely transitioned. You know, everything's turned to whatever was in the waters there. Now, that's the hair follicle shaft coming up through the top, you see it? It's got a flat side, which means it would be a curly-haired person, is what I've been told. Now, this is pretty obvious what this is, but it is 1,000 times bigger than a human hair. Human hair, they say, is 2/1000 of an inch wide. Now, I don't know if they're talking about this or the bulb. Let's say even 500, even 100 times. That would be a 500-foot tall person if he was a 5-footer. I mean, just think about it. This is good size. And my other ones were DNA tested, and that was like I said, that was a 3-foot long fingertip. This one here, that's 3 feet long from here to here. To the very end where it wraps around the front was 3 feet long. Fingernail, fingerprints, the whole 9 yards. So I think it's time to speak up to academia and say you gotta come to your senses here and start teaching reality. It's not fair to the kids, not fair at all. So I'm going to leave it at that note, but I think you should step up and start talking. Somebody's got to start talking. You see how long I've been talking, and I tried to be as nice as I could be. I was just because I knew I was going to hit this. You know, it's shocking. It's shocking as hell. And it was to everybody. And the truth is, I basically lost everybody in my life as I started talking about it, because they just thought this guy's going off his rocker. And most of them never came back. They still feel the same way. It's just that they're indoctrinated. Menticide means you cannot think for yourself. And the ones that can think for themselves realize they can't talk. Now, what a position to be in. I can't imagine being in that position where I can't talk knowing the truth. Because once you know the truth, there is no excuse. Okay, my friends, let's just end this on a semi-positive note. You cannot deny this. Now, to continue to deny this is really obstruction of justice. Call it anything you want, but it's not truth, and it's not real, and it's not fiduciary obligation, fulfilling the fiduciary obligations and living up to your moral and ethical responsibilities to look into something that has substantial proven evidence. Which I did. DNA, CAT scans, all of that stuff. They would accept that. They should. That's what they showed. Well, they don't even show that. So anyway, this is just a problem. Once you know the truth.
Once you know the truth, there is no excuse. Once you know the truth, there is no excuse.
I love you all.